All right, y'all, we are going to bring on our special guest for the night, Professor, Doctor of the Merritt variety.
I, at that point, didn't even realize how big The Last of Us was. I don't get out a lot. The thing that's been amazing to me doing some of the background on this is I had the impression that these kind of interactions between fungus and insects were relatively rare. Like, I actually have a set of notes. Like, there's some interesting ones in ants. There's a series of really interesting ones in flies, obviously. And then cicadas. But it's something like 2/3 of insect orders have examples of this kind of parasitism. It is just rampant. So you know this. You know that you're ahead of me on this.
Yeah, and each of them like have their own species of, let's say, like the cordyceps fungus that specializes on them. Right? So it's not even like that it's like a generalist that's just hanging out hitting all these insects, but it's like it's almost one-to-one in some cases.
Yeah, actually it might be. And so as I was reading through that, the thing that struck me was this system of parasitism and taking over the insect system has evolved, in this case, not 3 or 4 different times, but thousands potentially of times. And it really is very much different fungi in different lineages of insects. And it crosses huge amounts of phylogenetic space in both insects and fungi. So it's not like there are particular insects that are susceptible or there are particular fungi that are good at this. It's happened over and over again in different orders. So when I sent you the note and said, what should we chat about this time? The two that you sent me are really cool. So one is I saw you had an image up of the biomimic spore cannon, and then the other was these flower mimics, right? And there are a couple of interesting sort of unifying themes through this whole fungal thing. One, we've already said, like this parasitism is an incredibly successful lifestyle. And there are these fungal plant parasites just almost ubiquitous. Like, they certainly are far more widespread. A common theme through all of this is how are these parasites getting from A to B? So the cannon story is really cool because we're literally projecting spores, and we're shooting spores out. Okay, we'll go into spores. We'll dive into spores. There's some really cool things in the biomechanics of this. But there's some unifying ideas. So we've got these spore cannons, and then we've got these pseudo-flowers that are growing on plants. And when I first pulled them up, I was like, I want to read these because they look really cool. But they're actually doing exactly the same thing in 2 different mechanisms. In both cases, what has evolved is a way to get the spores spread through the environment. So in the one case, the fungi has infected an insect. It spreads through the insect. It changes the behavior of the insect in really, really, really scary ways. And then it spreads itself through the environment to infect more insects. So that's pretty straightforward. Like, okay, well, how is this evolved? It's evolved as a way to get spores from one insect to another insect and the insect as host. In the pseudo-flowers, the whole system has evolved to get spores from one plant to another plant. And it's using this insect as a delivery mechanism, but it's not infecting the insect, but it's doing exactly the same thing. It's spreading spores. And so in the pseudo-flower case, what's happening is the plant gets infected. The fungi changes the gene expression in the plant, which is exactly what it does in an insect. It changes the gene expression in the insects. In this case, instead of changing the behavior of the fly or the cicada or the ants, it changes a cell type in the plant to develop into what looks like a flower, these pseudo-flowers. At the same time, not always, but it often sterilizes the flower. So the plant flower no longer functions. There is now this pseudo-flower that smells like, looks like, tastes like a flower. A pollinator comes in, lands on it, picks up fungal spores, and then transmits that to another plant. And so all it is a different mechanism for getting spores spread through the environment and infecting a greater swath of individuals. So the selective pressure is how many things can you infect and spread. It's just these seemingly 2 very distinct. You've got cannons firing out of flies and pseudo-flowers sprouting out of stems, and they're doing exactly the same.
What is the potential achievable goal other than learning how it works? I guess, like, what is the translational use of something like this?
So one is to prevent the global apocalypse. I mean, we've all watched TV and we know it's coming. That's not true. So there are a couple of reasons. One, honestly, is disease infection. So the only way to stay ahead of the next global apocalypse or the global pandemic, the next plague, is to understand where these plagues come from. And so if we had spent more time studying the 1918 Spanish flu, so the last flu plague, we would have been better prepared for this flu plague. And, it's not a matter of, is there going to be a global infection of fungus? There are, and we need to understand how fungus infects and how they're spread. If nothing else, my favorite friend, coffee, may be extinct in 50 years because of coffee leaf rust, which is a fungal infection that spreads through essentially all commercially grown coffee.
So it's not just our health, but also our food. Anything that can tangentially affect our lives, understanding how it works, we can figure out an intervention to stop 100%. That spread.
Yeah. One of the reasons that we study these biological systems is to understand how we can use the engineering, and which is not a conscious engineering, but the way that biology has tackled a particular problem to understand things. And so it's a bit of a leap that understanding the biomechanics of spore spewing is going to help us understand something, but 100%, this is how we make advances. And so I was reading this and I'm like, this is it. They're describing it as a biomimetic structure. And I was like, that's really cool. I wonder what other biomimetic structures are out there that people have looked at. So Velcro pops up if you do that. But you have enough experience with an octopus that you can picture an octopus tentacle doing something like this. You've got a series of muscles that have fired in a coordinated fashion that have moved your hand in a rotation, right? And so what's happened is the muscles are pulling here, releasing here, pulling here, releasing here, and they're moving around a skeletal system that allows your hand to manipulate. Even just to make a fist, right? So you can picture an octopus tentacle doing that, swirling around. They do it all the time, right? How many bones are involved with that with an octopus?
None.
Absolutely zero. And so what they are is a muscular hydrostat, and it's a tube that's a fixed volume. And when you pull on that fixed volume, it becomes rigid. If you pull on that in a helical fashion, you can get it to rotate, right? And so we have one of those ourselves that we're using at the moment. I use all the time. Your tongue does very similar manipulations to your hand using absolutely no bones. So the way that we understand how an octopus tentacle moves, and it moves because it's got a series of muscles and connective tissues that are spun around it in a helical fashion that give it turgor and allow it to twist and move, exactly the way that your tongue works. So understanding how the biological world works allows us to understand how we as members of that biological world also function. What are the applications of this spore cannon? One, it's just another example of a biological solution to an interesting problem. Mm-hmm. One of the interesting things that I found looking at these different ways for spores to get from point A to point B are some intermediate steps that are in between there. It's an amazing system where there is a fungus that infects a plant. The fungus gets into the flower. The fungus extends the flowering time of that plant. And so a lot of these cases that we're looking at are parasitism. So the fungus takes over the insect. When we think of this idea of an insect being taken over by a fungus, there's some intermediate examples where instead of parasitism, it's mutualism. And so in this particular hummingbird case, the plants that are infected with the fungus flower for longer. And so the plant benefits because it has a longer period of time for hummingbirds to feed from it and disperse its pollen to other plants. So we think that these systems evolve through sort of intermediate steps. And one of the reasons that we have, one of the pieces of evidence to support that is we find these intermediate steps. So in this hummingbird flower example, it's not parasitism. The fungus is infecting the plant. It's causing that plant to flower longer, which actually has a benefit to the plant. The hummingbirds come in, pick up the pollen, they pick up the nectar, which they're feeding on. They pick up pollen, which they then disperse. And because the plant is flowering longer, they get more nectar, more time to get pollen. The plant benefits as well. All right. So
the plants benefit. So the pollen here is the original plant's pollen, right? The fig tree. And then, so what's the fungus getting out of it?
The fungus is getting dispersal. That's the whole game, is the hummingbird is picking up fungal spores and spreading them to other plants.
So it's both fungal spores and the pollen from the plant. So,
Yeah. So
which is different from the one that we were just like the paper we were talking about, where it's a because it sterilizes in some instances, right? The flower.
Right. And so this is an interesting case where the authors are making the argument that in many of these pseudo-flower cases, the plant itself is sterilized, and so it really is parasitism. But in some other cases, the plant isn't sterilized. And so there is a functioning flower that pollinators can go to and get nectar, but also get pollen and spread the pollen in a typical pollinator fashion. Or they can go to this pseudo flower, and the plant gets nothing from that, but the fungus thing gets spread to other plants. The different flavors of this system, in some cases, the pseudo flowers are sort of hyper flowers, and this is my terminology, not theirs, which is why it's sort of terrible, but it's sort of a hyper effective flower. These pseudo flowers can look more like flowers than flowers and become hyper effective flowers. And they do it sometimes because they look like flowers, they do it sometimes because they smell like flowers, and they do it sometimes because, although they don't look like flowers, the color pattern, or at least the refraction, is right. And so the pollinators go to the pseudo flower in preference to the real flowers.
There was a question earlier in the chat about that, is whose genes are causing that to happen with the pseudo flower? Is it the fungus manipulating the plant's gene to make it more flowery, or is there something in the fungus genome that also codes for some of these flowers or odors or what have you? There must be something, right, that it's machinery to hijack in one way or another.
Well, and so there's a really cool question, because you can imagine it going different ways. You can imagine that the fungal genome is, say, a set of gene expression regulators, and it turns on the expression of the wrong set of plant genes at the wrong place. Or you can imagine that the fungus is actually being expressed as like a fruiting body in place of the plant. And it ends up that there are probably examples of all of those. In at least one of the cases that I was looking at, what happens is the pseudoflower is made up of misexpressed plant tissue and fungal tissue. So different pieces of that pseudoflower are either fungal or plant. In that case, it's pretty obvious how we can get the spores out, because the fungal part of the pseudoflower is producing those spores, which are then spread when a pollinator comes in. If that pseudoflower was entirely plant, you'd have to have some kind of secondary system that allowed it to express spores. But that is what we see in the insects, where the insect is all insect, and then the fungus sprouts out of that insect.
It's a really elegant and beautiful system.
It's a nasty, terrible, horrible, really amazing system.
If it's hijacking the genetic machinery of the plant, how is it driving it to be that hyper-effective flower, both in the sense of, I guess, structure and then odor too?
So the fungus doesn't know anything, and the number of organisms that actually know anything are relatively small. We're pretty comfortable that we as human beings know things and make decisions. Even complex insect behaviors, it's unclear how much the insect knows what it's doing versus it is following a fixed set of patterns. So the fungus doesn't know anything. How does the fungus get it right? If the fungus doesn't get it right, it doesn't go on to the next generation. So a fungus that produces a pseudoflower that smells like a flower, or smells even more like a flower than a flower does, is more likely to reproduce than a fungus that smells like a fungus. And so you're going to get small changes over evolutionary time where that pseudoflower begins to smell more and more like a flower and less like a mushroom. But there is no conscious decision to do that. It's simply that in the rare mutation that makes that pseudoflower smell more flower-like, that's more successful. I think that at least in some of the cases, what's happening is a piece of the pseudoflower is a fungal flowering body. That's where the spores are going. A piece of the pseudoflower, other pieces of the pseudoflower are pieces of plant that are essentially cancers. So, I mean, cancer is just missed gene expression, right? So what's happening in this case is the fungus is causing misexpression of a series of genes and creating this pseudoflower, which you can really think of as a tumor, but it's a directed tumor, right? And so it's not just this mass, it's a mass that looks like, in some cases, petals or pieces of leaves or whatever part of that pseudoflower. Pirated regulation of genes. And so there'll be fungal tissue that's expressing fungal proteins, fungal genes, fungal proteins, and then they're going to be plant tissues that are expressing plant genes, plant proteins, but in the wrong place because they've been reprogrammed by the fungus.
And so that's an inherently cool thing to study that could apply to us too, whether it be to crops or disease, is that external variable of the fungus or what have you, hijacking genes with it from the host. So it's like a really deep way of studying that host pathogen interaction.
Absolutely. Yeah, it's super cool. I said gene misexpression. It's pirated expression. So the fungus is driving expression of a very specific set of genes in a very specific location, just not the typical location. Different pseudo-flowers have different characteristics. I think you had that image, right? We've got these 3 different types of flowers that some look more like flowers than others. And so they're going to be a different set of genes that are being pirated in different cases, where you're going to either, that's the one, where you're going to either look more or less like a flower, or you're going to smell more or less like a flower. So they're going to be a different set of genes. So different fungal plant interactions have created these pseudo-flowers in different ways. And so you've got a different set of genes in A, B, and C.
And so by studying, so when you study A, B, and C, they could be very, very different answers. And that gives you more identifying how this system works, right? Like of different ways that we can independently modify, like different, to get to the same answer.
That's right. And so this is actually surprisingly and frustratingly common. So I think we've talked about some of the work we do in my lab. So one of the things that we've been working on is we use nickel as a way to stress flies, and we look to see how resistant to nickel different lines of flies. So they're all Drosophila melanogaster, but they're essentially different families of Drosophila melanogaster is one way to think about it. And when we look across even a small group of lines, say like 10 lines of flies, we have nickel resistance or nickel sensitivity that varies by like an order of magnitude. So we've got really sensitive and really resistant nickel flies. And so an application of this is to say, okay, well, what genetic pathways are involved with metal resistance, so we can understand either how we are susceptible to metal contamination or how things like pests are susceptible to metal contamination in the environment. So one of the reasons that we use a model species is we can find the pathway for metal resistance, and then we understand how stress works, metal resistant works, that kind of thing. Even in this small set of 10 lines of flies, we know that 2 of our resistant lines have independent genetic pathways, because when we cross them, if it was the same pathway, you would get the same resistant fly. And in fact, what we get is a resistant, and then super resistant fly. So we're combining 2 different resistant pathways in this cross. So even in that small case, it looks like 2 distinct genetic pathways are involved for exactly the same characteristic.
So in theory, we could make these 2 mutations from, plant A, plant B into the same plant, and then it maybe even be with double resistant, let's say.
Yeah,
possibly.
But it's equally likely that when you cross them, even though it's resistant and resistant, what you might make is a hypersensitive fly, because there's nothing that says that these 2 pathways have to be additive. In fact, they could be directly combative, and those 2 mutations actually exacerbate rather than exaggerate each other. Given that we've got, so many examples of these, like, does it, what are the chances it always sterilizes it? Well, many cases it doesn't. And so what's happening then is the flower is still able to see. So there are different evolutionary paths that can be successful. So in one case, the plant is able to reproduce and grow on its own because it's typically there. In the other case, the parasitism, this co-option, is even the pirating of the system is so complete that the plant itself is just shut down. And so the plant is no longer effectively reproducing at all, and all of the energy is going into that fungus. You can imagine different successful strategies. And so this is actually the reasoning behind our understanding of how things like the plague have worked.
What is the benefit to the fungus to sterilize its host? Because if it sterilizes its host, like, how does it gonna then, right, eventually if it sterilizes all of its host, it's gone. It's like, right. what's the benefit?
So that's an interesting, and that's the balancing act. Like, if it is so effective at sterilizing the host that the host disappears, then that lineage dies out too. So and you mentioned there's a lot of 1-to-1. So there's a lot of specificity between the fungal species. It's not like the fungi are infecting lots of different plants. There's a fungus and a plant in the relationship. And so there's a balancing act that seems to have evolved where it's deadly enough, or it's nasty enough to suppress the flower but not drive the flower to extinction. The advantage is if the plant itself is not flowering, then no pollinators go to the real flowers. They all go to the pseudo-flowers. The disadvantage is you are really suppressing the fitness of that host, and parasites live by having hosts that are still successful.
And so then what would you say is the likelihood that it's going to sterilize a host? And then is there one question we always get when it comes to this interaction is what defense does the host have?
So everything's an arms race, and in the same time that the fungus is evolving through time and creating, say, more successful pseudo-flowers, if there's a plant that's resistant to fungal infection, it's going to be more successful. And so that's the arm race: fungal resistance, infection resistance versus more effective pseudo-flowers. So there's definitely a balancing act that goes between there. There is not one trajectory. This is a very chaotic system, and it's likely that in different lineages, you get different paths and different end results.
So it's not always that it's the host loses. There has to be a balancing act. And sometimes, like we've talked about with Chad, it's that Red Queen hypothesis where you're just running in place.
Yeah.
Thank you for taking the time, your knowledge, your wisdom, imparting on us. We appreciate you, my friend.
It's great to see you and great to chat with everybody. I'll see you next month.
See you. Bye, Thomas.